Every night, as darkness falls across the globe, billions of humans engage in one of the most peculiar behaviors in all of nature. We lie down, close our eyes, and surrender to unconsciousness for approximately 8 hours. During this time, we become vulnerable, unproductive, and disconnected from the waking world. For early humans living among predators and competing tribes, this nightly ritual seemed like evolutionary suicide. Yet sleep persists across virtually every animal species on Earth, from tiny fruit flies to massive blue whales, suggesting it serves purposes so critical that evolution has preserved it despite its obvious dangers. The scientific study of sleep is surprisingly young. While humans have wondered about dreams and sleep for millennia, rigorous scientific investigation only began in earnest during the 20th century. The discovery of rapid eye movement sleep in 1953 by Eugene Azarinsky and Nathaniel Kale Yitman opened the floodgates to modern sleep research. Since then, scientists have developed numerous theories to explain why we sleep, what happens during sleep, and how sleep affects our health, cognition, and longevity. These theories range from the molecular level, examining individual neurons and chemical processes, to the evolutionary scale, considering how sleep emerged over millions of years of natural selection. Understanding sleep theories matters more today than ever before. Modern society faces an unprecedented sleep crisis. Artificial lighting, digital devices, shift work, and cultural attitudes that prize productivity over rest have created a world where chronic sleep deprivation has become normalized. According to sleep researchers, this epidemic of insufficient sleep contributes to obesity, diabetes, cardiovascular disease, depression, reduced immune function, and impaired cognitive performance. The economic costs alone run into hundreds of billions of dollars annually in lost productivity, medical expenses, and accidents. On an individual level, understanding what sleep does for us can motivate better sleep habits and help us recognize when our relationship with sleep has gone awry. This script explores the major scientific theories that attempt to explain the phenomenon of sleep. Each theory offers a different lens through which to view our nightly unconsciousness, and together they paint a picture of sleep as a remarkably complex, multifaceted biological process. Some theories focus on evolutionary origins, asking why sleep emerged in the first place. Others concentrate on specific functions like memory consolidation or cellular repair. Still others examine the regulatory mechanisms that govern when we feel sleepy and when we feel alert. Modern neuroscience has revealed that sleep is not a single uniform state, but rather a dynamic process involving multiple stages, each with its own characteristics and purposes. As we journey through these theories, we'll discover that sleep is far more than simply rest. It's a time when the brain reorganizes itself, when memories are sorted and strengthened, when cellular damage is repaired, and when metabolic systems are recalibrated. We'll learn about the recently discovered Glymphatic System, which flushes toxic waste products from the brain while we sleep. We'll explore how the same evolutionary pressures that shaped our ancestors' sleep patterns continue to influence how modern humans sleep today. And we'll examine how understanding these theories can help us optimize our own sleep for better health, performance, and well-being. The beauty of sleep science lies in its interdisciplinary nature. Unraveling sleep's mysteries requires insights from neuroscience, evolutionary biology, molecular biology, psychology, endocrinology, and more. Each discipline contributes pieces to the puzzle, and the theories we'll explore represent attempts to assemble those pieces into coherent explanations. While no single theory yet provides a complete picture of sleep, together they offer profound insights into one of the most fundamental aspects of human existence. As we stand at the frontier of sleep research, with new discoveries emerging regularly, we're closer than ever to answering the ancient question, why do we sleep? When considering any biological behavior as universal in time minus consuming as sleep, evolutionary biologists instinctively ask what evolutionary pressures lead to this behavior. What advantages did sleep confer on our ancestors that outweighed its obvious costs? The evolutionary theory of sleep encompasses several interconnected hypotheses that attempt to answer these questions by examining sleep through the lens of natural selection. These theories suggest that sleep emerged and persisted because it enhanced survival and reproductive success in ancestral environments, even though the specific mechanisms by which it did so may have varied across species and ecological niches. The adaptive inactivity theory, also called the immobilization theory, proposes that sleep evolved as a survival strategy that kept animals safe during times when they were most vulnerable when being active, provided minimal benefits. Consider a small rodent that relies on vision to navigate and find food. During the pitch-minus black nights of the pre-minus electric world, attempting to forage would be dangerous and inefficient. This rodent would be vulnerable to nocturnal predators with superior night vision, might injure itself moving through darkness, and would expend precious energy with little chance of obtaining food. Natural selection would therefore favor individuals who possessed an innate drive to become inactive and hide during nighttime hours. Sleep, in this view, is essentially an enforced inactivity that prevents animals from engaging in risky behavior during support sometimes. This theory finds support in the tremendous variation of sleep patterns across species. Animals sleep when it makes evolutionary sense for them to do so, based on their ecological niche, predation risk, and sensory capabilities. Herbivores like horses and elephants sleep relatively little, just 2 to 4 hours daily, because as large prey animals, they must remain vigilant for predators, and because their low-calorie diet requires nearly constant foraging. Conversely, apex predators like lions sleep up to 20 hours per day, secure in their position atop the food chain and with energy-rich diets that don't require constant hunting. Aquatic mammals like dolphins and whales have developed truly remarkable adaptations, sleeping with one brain hemisphere at a time so they can continue surfacing to breathe. Each species' sleep pattern reflects its unique evolutionary history and ecological pressures. The energy conservation hypothesis offers a complementary evolutionary explanation, focusing specifically on metabolic efficiency. Sleep reduces metabolic rate, sometimes by as much as 10 to 15%, conserving valuable energy resources. For animals living in environments where food is scarce or unpredictable, these savings could mean the difference between survival and starvation. During sleep, body temperature drops slightly, muscle activity decreases dramatically, and various physiological processes slow down. These changes reduce overall energy expenditure, allowing animals to maintain their energy balance more easily. This theory is particularly compelling when considering hibernation, which can be viewed as an extreme form of sleep that allows animals to survive months without eating by drastically reducing metabolic demands. Mathematical models support the energy conservation hypothesis. Researchers have calculated that the metabolic savings from sleep, while modest in absolute terms, could be significant over evolutionary timescales when resources were limited. For a small mammal, the energy saved during sleep might equal the calories obtained from an entire foraging bout. Over weeks, months, and years, these savings accumulate substantially. Moreover, the energy conservation benefit would have been particularly important during periods of scarcity, such as harsh winters or droughts, when the animals that slept more efficiently would have better survived to reproduce. The predator avoidance component of evolutionary sleep theory highlights another crucial survival benefit. For many animals, particularly prey species, being active increases predation risk. Movement attracts attention, and predators often have sensory systems exquisitely tuned to detect motion. By remaining still and hidden during sleep, animals reduce their detectability. Furthermore, sleep often occurs in protected locations, burrows, nests, or other refuges where predators cannot easily reach. The depth of sleep varies by species according to predation risk, with prey animals typically being lighter sleepers that can awaken quickly when threatened, while predators enjoy deeper, more prolonged sleep periods. This variation suggests that natural selection has fine-tuned sleep characteristics to match each specific ecological circumstances. Interestingly, sleep itself may create evolutionary pressure for predator vigilance, generating an arms race between predators and prey. As prey animals evolve to sleep for energy conservation or other benefits, predators face selection pressure to hunt during these vulnerable periods. This, in turn, drove the evolution of lighter sleep, sentinel behavior where some group members stay awake while others sleep, and the remarkable unihemispheric sleep of marine mammals and some birds. The evolutionary dance between predators and prey has thus shaped not just whether animals sleep, but how they sleep, when they sleep, and how deeply they sleep. Cross-species comparisons reveal fascinating patterns that illuminate sleep's evolutionary origins. All mammals sleep as do all birds, suggesting that sleep evolved at least by the time these lineages diverged from reptiles over 200 million years ago. Even simpler organisms show sleep-like states. The fruit fly Drosophila melanolitus exhibits periods of inactivity with reduced responsiveness to stimuli, increased arousal threshold, and a rebound effect if deprived of rest. These similarities to mammalian sleep suggest that fundamental sleep mechanisms may be ancient, perhaps dating back hundreds of millions of years to early animal evolution. If even organisms with relatively simple nervous systems display sleep-like behaviors, this implies that sleep serves purposes so fundamental that evolution discovered and preserved them very early in animal history. The persistence of sleep across such vast evolutionary distances and diverse ecological contexts suggests that sleep serves multiple essential functions. And exclusively adaptive inactivity function seems insufficient to explain why animals with vastly different lifestyles, from dolphins to bats to humans, all maintain complex sleep architecture with multiple stages. Similarly, pure energy conservation doesn't explain why sleep deprivation causes such severe impairments beyond simple fatigue. Modern evolutionary theorists generally conclude that sleep likely evolved initially for adaptive inactivity or energy conservation, but then was co-opted for additional crucial functions related to brain and body maintenance. Once animals were necessarily inactive for certain periods, evolution could leverage that downtime for other essential processes that work better when the organism is not responding to environmental stimuli. This evolutionary perspective helps explain some puzzling aspects of human sleep. Our ancestors lived in environments dramatically different from modern industrialized societies. They experienced natural light cycles without artificial illumination, faced genuine predation risks, endured periods of food scarcity, and lived in close, knit social groups. Our sleep systems evolved under these conditions, which explains why we struggle with artificial light, why we sleep better in cool, dark, quiet environments, and why social support and security promote better sleep. Understanding sleep's evolutionary origins can thus guide interventions to improve modern sleep by recreating aspects of ancestral sleep environments, such as maintaining darkness, establishing regular schedules synchronized with natural light cycles, and creating safe, comfortable sleeping spaces. Among the most intuitively appealing explanations for sleep is the restorative theory, which proposes that sleep provides essential time for the body and brain to repair damage accumulated during waking hours. This theory resonates with our subjective experience. We feel tired after sustained activity and refreshed after good sleep. We recognize that illness increases our sleep need, and we instinctively seek rest when injured or stressed. The restorative theory suggests that these intuitions reflect genuine biological processes, with sleep serving as a crucial period for cellular repair, tissue growth, immune system strengthening, and restoration of depleted resources. At the cellular level, sleep facilitates numerous repair and maintenance processes. During waking hours, metabolic activity generates reactive oxygen species and other potentially damaging byproducts. These molecules can damage cellular components including DNA, proteins, and lipids. while cells possess repair mechanisms that function continuously. Many of these processes work more efficiently during sleep when reduced metabolic demands free up resources for maintenance rather than immediate functional needs. Research has demonstrated that genes involved in cellular repair and protein synthesis show increased expression during sleep, suggesting that sleep creates an optimal environment for cellular housekeeping. Protein synthesis particularly benefits from sleep. Proteins are the workhorses of cellular machinery serving structural roles, catalyzing biochemical reactions, and enabling cellular communication. During waking activity, proteins experience wear and damage. Sleep allows for increased production of new proteins to replace damaged ones. Studies have shown that sleep deprivation impairs protein synthesis in multiple brain regions and peripheral tissues. This impairment could help explain why chronic sleep restriction leads to various health problems, as cells cannot adequately maintain and repair their protein machinery. The growth hormone surge during deep sleep further supports this restorative function, as growth hormone stimulates protein synthesis, cell reproduction, and tissue repair throughout the body. Muscle tissue provides a clear example of sleep's restorative role. Athletes and anyone engaging in physical training understand that actual muscle growth occurs during rest, not during exercise itself. Exercise creates microscopic damage to muscle fibers, and the body responds by repairing and reinforcing these fibers, making them stronger. This process requires protein synthesis, which occurs predominantly during sleep when growth hormone levels peak. Sleep deprivation impairs athletic recovery, reduces muscle protein synthesis, and increases perceived exertion during subsequent exercise. Elite athletes often prioritize sleep as essential to their training regimens, recognizing that inadequate rest undermines their physical development and performance. The immune system demonstrates perhaps the most dramatic connection between sleep and restoration. During sleep, the immune system releases various cytokines, proteins that help fight infection and inflammation. Certain cytokines need to increase during infection or inflammation, and sleep deprivation decreases production of these protective cytokines. Sleep also enhances production and function of T cells, which are critical for fighting viral infections and cancer cells. Research has shown that people who consistently get insufficient sleep are more susceptible to common infections like colds and flu. In one striking experiment, researchers exposed volunteers to cold viruses and found that those sleeping less than 7 hours per night were approximately 3 times more likely to develop illness compared to those sleeping 8 or more hours. Sleep's role in immune function extends beyond infection resistance to include inflammatory regulation. Chronic sleep deprivation promotes systemic inflammation, with increased levels of inflammatory markers like C- reactive protein and interleukin-6. This inflammatory state contributes to numerous chronic diseases including cardiovascular disease, diabetes, and neurodegenerative disorders. The relationship appears bidirectional. Poor sleep increases inflammation, and inflammation disrupts sleep quality, potentially creating a vicious cycle. Understanding this connection highlights sleep as a crucial component of preventive healthcare. as improving sleep may reduce chronic disease risk through multiple mechanisms, including inflammation reduction. Metabolic restoration represents another key aspect of sleep's restorative function. During sleep, the body rebalances various metabolic systems and replenishes depleted energy stores. Glycogen, the storage form of glucose, gets replenished in the brain during sleep. The brain consumes enormous amounts of energy relative to its size, using approximately 20% of the body's total energy despite representing only 2% of body weight. This intense metabolic activity depletes energy stores that must be restored. Sleep provides an ideal time for this replenishment because reduced cognitive demands during sleep lower energy consumption, allowing restoration to exceed depletion. The restorative theory also encompasses cardiovascular recovery. During non-REM sleep, particularly deep sleep, blood pressure drops significantly, heart rate slows, and the cardiovascular system gets a reprieve from the demands of supporting active metabolism. This nightly blood pressure dip appears crucial for cardiovascular health. People who don't experience this nighttime dip, often due to sleep disorders or disrupted sleep, face increased risk for hypertension, heart attack, and stroke. Sleep thus provides essential recovery time for the heart and blood vessels, allowing these systems to operate below their daytime capacity and undergo repair processes. Hormonal regulation provides further evidence for sleep's restorative function. The endocrine system follows circadian patterns synchronized with sleep-wake cycles, with various hormones showing distinct profiles across the 20-hour day. Growth hormone, as mentioned earlier, surges during deep sleep, facilitating tissue growth and repair. Cortisol, a stress hormone, follows an inverse pattern, dropping to its lowest levels during early nighttime, sleep before rising toward morning. Leptin and ghrelin, hormones regulating appetite and metabolism, also show sleep-dependent patterns. Sleep deprivation disrupts these hormonal rhythms, contributing to metabolic dysregulation, increased appetite, weight gain, and reduced glucose tolerance. The specific restorative processes occurring during different sleep stages suggests that sleep architecture evolved to facilitate multiple distinct restoration functions. Deep non-REM sleep, characterized by slow brain waves, appears particularly important for physical restoration, with maximum growth hormone secretion and extensive bodily repair processes. REM sleep, characterized by rapid eye movements and vivid dreams, may focus more on brain restoration and emotional processing, though it also supports important physiological functions. The cycling through different sleep stages ensures that all necessary restoration occurs, which may explain why both sleep quantity and quality matter for health outcomes. At the organismal level, the restorative theory helps explain numerous clinical observations. Illness increases sleep need because fighting infection and healing tissue demand additional restoration time. Injuries promote longer, deeper sleep to facilitate healing. Psychological stress increases sleep need because stress hormones damage tissues and deplete resources. Conversely, chronic sleep restriction creates a state of incomplete restoration, where damage accumulates faster than repair processes can address it, leading to the myriad health consequences associated with insufficient sleep. This framework positions sleep not as passive downtime but as active restoration, an essential biological process as important as nutrition or exercise for maintaining health. Perhaps the most extensively researched modern sleep theory focuses on sleep's critical role in learning, memory, and brain plasticity. Over the past several decades, an enormous body of evidence has accumulated demonstrating that sleep is not merely passive rest, but an active state during which the brain processes, consolidates, and integrates information acquired during waking hours. This theory positions sleep as essential for cognitive function, suggesting that one of sleep's primary purposes is to optimize neural networks, strengthen important memories while pruning irrelevant ones, and prepare the brain for new learning. Memory consolidation, the process by which newly acquired information transitions from fragile short-term storage to stable long-term memory, represents one of sleep's most well-established functions. When we learn something new, whether factual information, a motor skill, or a life experience, that information initially exists in a relatively unstable state. Without consolidation, memories fade rapidly. Early research showed that people who sleep after learning retain information better than those who remain awake for an equivalent period. This wasn't simply due to reduced interference. Sleep actively strengthened memory traces through neurobiologic processes occurring during specific sleep stages. Different types of memories benefit from different sleep stages, suggesting specialized consolidation mechanisms. Declarative memories, the conscious memories of facts and events like remembering a phone number or what you had for breakfast, benefit particularly from non-REM sleep, especially the slow-wave sleep state. During slow-wave sleep, the hippocampus, which serves as a temporary storage site for new memories, systematically replays patterns of activity that occurred during learning. These reactivations, detectable through brain imaging and electrophysiology, appear to drive the transfer of information from the hippocampus to the neocortex for long-term storage. With each replay, synaptic connections between relevant neurons strengthen, stabilizing the memory. Procedural memories, the unconscious memories of skills and procedures like riding a bicycle or playing a musical instrument, show particularly strong dependence on REM sleep. Studies of musicians learning new pieces, athletes acquiring motor skills, and participants mastering laboratory tasks consistently demonstrate that REM sleep enhances procedural learning. During REM sleep, motor cortex and subcortical motor regions show increased activity and plasticity. The brain appears to be rehearsing and refining motor programs, optimizing movement sequences without the need for actual physical practice. This offline rehearsal explains why we sometimes wake up better at a skill than when we went to bed, even without additional practice. The synaptic homeostasis hypothesis, proposed by neuroscientist Giulio Tononi and Chiara Surelli, offers a compelling framework for understanding sleep's role in brain plasticity. This theory suggests that during waking hours, learning and experience progressively strengthens synaptic connections between neurons. While this strengthening enables learning and memory, it cannot continue indefinitely. Synapses consume energy in space, and continual strengthening would eventually exhaust the brain's capacity. The synaptic homeostasis hypothesis proposes that sleep, particularly slow minus-wave sleep, serves to downscale synaptic strength overall, while preserving relative differences between synapses. This process, called synaptic renormalization, maintains the brain's ability to learn by preventing saturation while keeping important information encoded in the pattern of synaptic strengths. Evidence supporting synaptic homeostasis comes from multiple sources. Brain imaging studies show that markers of synaptic strength increase during waking hours and decrease during sleep. Molecular markers of synaptic size and strength follow similar patterns. Computer simulations demonstrate that networks implementing synaptic homeostasis show enhanced learning capacity and memory stability. The theory elegantly explains several sleep phenomena: why we need sleep after intense learning, why sleep deprivation impairs learning and memory, and why different sleep stages might serve distinct but complementary functions in memory processing. Sleep doesn't simply preserve memories unchanged, it transforms them through active integration and reorganization. Research has revealed that sleep promotes insight and creative problem-solving by enabling the brain to discover hidden patterns and relationships between disparate pieces of information. The famous cases of scientific discoveries emerging from dreams, such as August K. Ekule's realization of the benzene ring structure, may reflect genuine sleep-dependent cognitive processes. Modern experiments confirm that people are more likely to gain insight into rule-based problems after equivalent waking periods, suggesting that sleep facilitates the abstraction of general principles from specific examples. Memory consolidation during sleep shows remarkable selectivity. The brain doesn't indiscriminately strengthen all memories, rather, it prioritizes information tagged as important. Memories associated with reward, emotional significance, or future relevance receive preferential consolidation. The selectivity makes evolutionary sense limited neural resources should be devoted to remembering useful information while forgetting trivial details. Recent research has even demonstrated that providing specific cues during sleep can enhance consolidation of associated memories, a technique called targeted memory reactivation. In these experiments, playing out of those sounds previously associated with learned information during sleep strengthens those specific memories, demonstrating that sleep-dependent consolidation can be externally influenced. The neural mechanisms underlying sleep-dependent memory consolidation involve coordinated activity across multiple brain regions. During slow -wave sleep, a characteristic pattern emerges where cortical slow oscillations, thalamic spindles, and hippocampal sharp -wave ripples occur in temporal coordination. These coordinated events, sometimes called the triple minus phase coupling, appear to create optimal conditions for transferring information from hippocampus to cortex. During these moments, the hippocampus replays learned information. Thalamic spindles mediate information flow, and cortical slow oscillations create windows of heightened plasticity for receiving and encoding the transferred information. This intricate coordination represents one of the brain's most elegant computational processes. Sleep's role in emotional memory processing deserves special attention. REM sleep, with its associated high levels of acetylcholine and low levels of norepinephrine, may provide optimal conditions for processing emotional experiences. During REM sleep, emotional memories can be reactivated and reconsolidated in a numerical environment that weakens the emotional charge, while preserving the factual content. This process may explain why sleeping on it helps process difficult emotional experiences, why dream content often has emotional themes, and why REM sleep disruption is associated with mood disorders. The selective processing of emotional content during REM sleep might serve an adaptive function by helping organisms learn from emotional experiences without being overwhelmed by them. Practical implications of the memory consolidation theory are substantial. Students would benefit more from distributed learning with sleep between study sessions than from cramming without sleep. Athletes should prioritize sleep following training sessions to maximize skill acquisition. Anyone learning complex material, from languages to professional skills, should recognize sleep as an essential component of the learning process, rather than time that could be spent practicing. These insights challenge cultural attitudes that treat sleep as wasted time, revealing instead that sleep is productive time, during which crucial cognitive work occurs. Understanding sleep's role in memory and learning transforms how we approach education, skill development, and cognitive performance. One of the most exciting recent discoveries in sleep neuroscience is the Glymphatic System, a waste clearance mechanism that operates primarily during sleep. Announced in 2012 by researchers led by Amick and Niedergaard at the University of Rochester. This discovery revolutionized our understanding of sleep's restorative functions and provided a concrete mechanism linking inadequate sleep to neurodegenerative diseases. The Glymphatic system represents a striking example of how sleep serves specific, identifiable biological purposes rather than being merely passive downtime. The brain faces a unique waste disposal challenge. Unlike other organs, the brain lacks a traditional lymphatic system for removing metabolic waste products. Yet brain tissue is metabolically incredibly active, continuously producing waste products that could accumulate to toxic levels if not removed. For decades, scientists puzzled over how the brain cleared these waste products. The glymphatic system provided the answer: a network of paraventricular channels through which cerebrospinous fluid flows, flushing interstitial fluid and dissolved waste products from brain tissue into the venous system for disposal. The name glymphatic combines glia, the supportive cells in the brain that help drive the system, with lymphatic, reflecting its waste minus clearance function analogous to the body's lymphatic system. The system works through several coordinated mechanisms. Cerebrospinous fluid enters the brain along pair of minus arterial spaces surrounding blood vessels. This fluid then flows into the interstitial space between brain cells, driven partly by arterial pulsion and partly by aquaportin minus for water channels on astrocyte and feet. As cerebrospinous fluid moves through interstitial space, it collects metabolic waste products including proteins, cellular debris, and excess neurotransmitters. Finally, this waste minus laden fluid exits a long pair minus venous spaces, eventually draining into the lymphatic vessels and bloodstream for systemic clearance. The critical discovery regarding sleep is that lymphatic clearance increases dramatically during sleep compared to wakefulness. Studies using real minus time imaging in living animals demonstrated that lymphatic flow increases approximately 10 minus fold during sleep. This enhancement occurs because the interstitial space expands during sleep, growing by approximately 60%, which dramatically increases fluid movement and waste removal efficiency. Upon waking, the interstitial space contracts, and lymphatic clearance decreases substantially. This sleep minus dependent change suggests that brain waste clearance is one of sleep's fundamental purposes, providing a compelling reason why animals must spend significant time in a sleep minus-like state. Among the substances cleared by the Glymphatic System, beta minus amyloid has received particular attention because of its connection to Alzheimer's disease. Beta minus amyloid is a protein fragment that naturally forms in the brain through normal metabolic processes. In Alzheimer's disease, beta minus amyloid accumulates into plaques that damage neurons and contribute to cognitive decline. The glymphatic system clears beta minus amyloid from the brain, and this clearance happens predominantly during sleep. Studies have shown that even one night of sleep deprivation increases brain beta minus amyloid levels, suggesting that chronic insufficient sleep could contribute to beta minus amyloid accumulation over time. This finding provides a plausible mechanism linking poor sleep to increased Alzheimer's risk, a connection supported by numerous epidemiological studies. The positioning and sleep architecture relationship also matters for lymphatic function. Studies suggest that lateral sleep positions may enhance lymphatic clearance compared to supine or prone positions, possibly because gravitational forces, anatomical factors affect cerebrospinous fluid flow dynamics. Additionally, different sleep stages show varying levels of lymphatic activity, with deep non-REM sleep appearing particularly important for waste clearance. This stage dependence suggests that both sleep quantity and quality matter for maintaining healthy lymphatic function, as fragmented sleep that reduces deep sleep time might impair waste clearance, even if total sleep duration seems adequate. The glymphatic system clears numerous other potentially toxic substances beyond beta minus amyloid. Tau protein, another protein associated with Alzheimer's disease and other neurodegenerative conditions, gets cleared through lymphatic pathways. Lactate, a metabolic byproduct that accumulates during waking neural activity, requires clearance to maintain healthy brain function. Various neurotransmitters and their metabolites also get removed. Even the clearance of extracellular potassium, which accumulates during neural activity and must be removed to maintain proper neuronal excitability, depends partly on glymphatic mechanisms. The system thus serves broad waste management functions essential for maintaining healthy brain chemistry and preventing toxic accumulation of numerous substances. Factors that impair glymphatic function could therefore contribute to neurodegenerative disease and cognitive decline. Aging appears to reduce glymphatic efficiency, which might partly explain increasing dementia risk with age. Traumatic brain injury impairs glymphatic function, potentially explaining why head trauma increases later neurodegenerative disease risk. Certain genetic factors affect aquaportin minus for expression and localization, potentially influencing individual differences in glymphatic efficiency and neurodegenerational susceptibility. Cardiovascular disease and hypertension can affect arterial pulsing that helps doctor glymphatic flow. Even alcohol consumption, while promoting sleep onset, disrupts sleep architecture and may impair glymphatic clearance, possibly contributing to alcohol's neurotoxic effect. The glymphatic system discovery has profound implications for understanding sleep disorders and their health consequences. Sleep apnea, characterized by repeated breathing cessation during sleep, fragments sleep architecture and reduces deep sleep time. This disruption likely impairs glymphatic clearance, which could help explain the increased dementia risk associated with untreated sleep apnea. Chronic insomnia, by reducing total sleep time and possibly affecting sleep depth, might chronically impair waste clearance. Even the sleep disruption common in shift work or jet lag could have cumulative effects on brain health through impaired glymphatic function. These connections emphasize that sleep disorders are not merely inconvenient but potentially serious medical conditions with long-term neurological consequences. Research into the glymphatic system continues rapidly, with many questions still unanswered. Scientists are investigating whether pharmacological or behavioral interventions can enhance glymphatic clearance. They're examining how other factors like diet, exercise, and medications affect glymphatic function. They're exploring whether glymphatic imaging could serve as an early biomarker for neurodegenerative disease, potentially enabling earlier intervention. And they're investigating whether similar systems exist in peripheral organs, which could revolutionize understanding of how the body clears waste products throughout the organism. The glymphatic system provides a concrete, mechanistic answer to the question, why do we sleep? At least one essential purpose is brain waste clearance, a process that cannot occur efficiently during waking activity. This discovery moves sleep neuroscience beyond correlational observations toward understanding specific biological mechanisms. It also reinforces the importance of adequate sleep for long-term brain health, suggesting that chronic sleep deficiency might not just impair immediate cognitive function, but also increase risk for serious neurodegenerative diseases later in life. For anyone concerned about maintaining cognitive function and brain health as they age, protecting sleep becomes not optional, but essential, as critical as diet and exercise for healthy aging. Understanding when we feel alert requires examining the mechanisms that regulate our sleep minus wake cycles. The 2 process model, first proposed by Alexander Borbely in 1982, remains the dominant framework for understanding sleep regulation. This elegant model suggests that two independent processes interact to determine sleepiness and alertness: process S, representing sleep homeostasis or sleep pressure, and process C, representing the circadian rhythm or internal body clock. The interplay between these two processes explains patterns of sleep and wake signals under normal circumstances and helps predict the effects of sleep deprivation, shift work, jet lag, and various sleep disorders. Processes. The homeostatic sleep doctrine operates according to a simple principle: the longer you stay awake, the stronger your urge to sleep becomes. During waking hours, sleep pressure gradually builds, creating an increasing drive towards sleep. Once you fall asleep, this pressure dissipates, decreasing across the sleep period. Upon waking, pressure is at its minimum, then begins accumulating again. This process functions as a negative feedback system, ensuring that extended wake-up signals is balanced by adequate sleep. The rate at which sleep pressure builds and dissipates can be measured physiologically through slow wave activity during sleep, with higher slow wave activity reflecting greater prior sleep pressure. The molecular basis of processes involves adenosine, a nucleoside that accumulates in the brain during waking activity. Neurons continuously produce and release adenosine as a byproduct of energy metabolism. The longer neurons remain active, the more adenosine accumulates in extracellular spaces. Adenosine binds to receptors on neurons, particularly A1 receptors, which inhibit neural activity and promote sleepiness. This mechanism creates a direct link between metabolic activity and sleep pressure. More neural activity produces more adenosine, generating stronger sleep drive. During sleep, especially slow wave sleep, adenosine levels decrease as the molecule is cleared from the brain, reducing sleep pressure and allowing wake signals to return. Caffeine, the world's most popular psycho,